We'll get started. So I will talk a little bit about the data visualization and data exploration capabilities that we've built into the Hubmap data portal. And before I get started, I really want to begin with acknowledging the team behind the work, the people that are working with me on this project at Harvard Medical School. You see the names listed here. So we have a fairly large group of people who are contributing to making the kind of functionality possible that I'll show you in a couple of minutes. So of course I want to acknowledge the funding of the NIH Common Fund through the Hubmap program, which is really an incredibly exciting project, I think from a data visualization point of view, which is really at the heart of my research interests, because of the enormous richness of the data sets, the heterogeneity, the many different modalities that we need to integrate. And so, as you will see in the next couple of minutes, first of all, in this data portal, of course, one of the most important features of a data portal is to guide you to the data. So searching for data, discovering data sets is critical. I want to also talk a little bit about how we manage the provenance through the user interfaces in the data portal and how you can explore data and the relationships between different data sets as well as samples and donors. Of course, I'm going to give you an overview of some of the visualization tools that are available in the data portal as of now, and also give you a quick preview of some of the functionality that will be built into the data portal in the next coming couple of months. So this is what the homepage looks like right now if you were to go there. I didn't include a URL, but if you have good eyes, you will very quickly figure out what the URL for the Hubmap data portal is. And this bar chart that you see here is just fake data at the moment, but this would give you an overview of the available data sets across different data types. And, of course, we have some pointers to other related resources, Twitter stream incorporated here, and a very, very concise overview of how Hubmap is collecting data and then processing it. So let's start by talking a little bit about discovery of data sets in the portal. And there are no surprises. We have metadata for donors, and you can just browse donors using a faceted browsing interface, very, very standard. We have a couple of facets included here right now, such as age, race, and ethnicity, BMI of the donor, as well as gender. And I want to emphasize this is indeed gender and not sex. Because this is how this information is being reported to us. So there are, as an overview, this is currently, as it looks today, we have not released the portal, so it's incomplete, but by the time the data release will happen, this table will be complete and have all the metadata for all donors. You can do the same for samples. You can, filter by organ type or tissue specimen type. You can also use the donor level facets such as, donor, gender, age, etc., to filter organs here as well. And finally, we have that functionality for data sets too. So in data sets, you can search by data set type, again, organ, specimen type, and any of the donor level attributes. And here, for example, I've selected the 10x genomics data sets that were processed through the Salmon pipeline that Ziv just explained. And as you can see here, currently there are 12 such data sets that are in the system, and they're for various organs, spleen, thymus, lymph node. And let's just take a look at one of these data sets and see what we find in there. So I want to go and give you an overview of the data set details that you would find in the data portal. So when you open this data set, the first thing that probably jumps right into your eyes is the visualization of the data. But before we get to that, I just want to point out, there's sort of a standard header. Every entity in Hubmap, these are data sets, donors, samples, have a Hubmap ID that you see here at the top. And those are also ultimately what would be turned into a DOI if those were published at some point. And as we take a look here, scroll down a little bit, here's a visualization of this 10x data set as it was processed through Salmon. There's a UMAP projection of the data. You see there's a clustering of the cells. And in this case, I didn't even expand this out because the clustering that the pipeline does will simply assign cells to individual clusters, name the clusters like 0 through X, where X is the number of clusters, and that's it. So that is not particularly informative, but nonetheless, we provide this information right here and display it, and you can explore. You'll see this in a moment, how that will look like. So scrolling down a little bit further, there's provenance. We'll come back to that in a moment. There's this provenance table view, essentially, which would give you an overview of like how this data set was generated all the way, going back all the way to the donor that provided the original sample. So in this case, you see there was a 10x data set, like essentially raw data that was uploaded by the submitting center, in this case, the University of Florida Tissue Mapping Center. And then you can check the sample provenance. So there was an organ, a whole spleen, and there was a piece taken out of that spleen, and then there was some single cells or cryopreserved, and that was then the input for the assay. We'll get to that in a moment. You can also see some information about the donor, and of course you can directly navigate to the detailed pages of these entities. Scrolling a little bit further down, you also have a little file browser where you can access any of the results, or at least a subset of the results that we consider relevant for immediate access through the website. You see, we not only list the file names and the size, there's also a description of what each of these files contains, so you don't have to download it and open it and start wondering what it might be. We have also a link to the Globus data portal. That's a system that is essentially managing all of the data, the permissions, et cetera, that's sitting behind the data portal. So you typically are not exposed to that as an end user. But if you want to use any bulk download functionalities and advanced data transfer, you can do this through Globus. Now, if you click on a file to download it, you first have to agree to our data usage agreement. And once you've done that, you can grab files directly from the website through the web browser. In this case, it's just a PDF of that same projection that I showed you in the interactive visualization tool earlier. Now, in this case, I went back and to the dataset browsing page and selected all those codex data sets that were processed through the Cytokit SPRM pipeline that Ziv mentioned earlier. And in this case, we have 6 such data sets that have a QA status, meaning the pipelines completed successfully and there were no errors. You also see, if you take a very close look, there are 5 data sets that failed somehow during the analysis. Again, we're still processing the data, it's not final. So these kind of things are not surprising. But let's take a look at one of these codex data sets. And Mike mentioned it earlier, what codex can do. And I want to show you what that looks like in a data portal. So here is a codex data set was processed using the pipeline that Ziv described earlier. And again, the first thing that you see is a visualization. This is for in the design of the data portal, in the user interface for the data portal, one of the driving principles behind the design is that we want users to get to the data and being able to take a look at the data with as few clicks as possible. So we're trying to incorporate visualizations for any data type that we will have in the data portal. Right now, these exist for various single cell sequencing assays, some particular single cell RNA-seq assays. They exist for imaging assays, and they will also exist for single cell ATAC-seq and other approaches. So in this case, what I want to do is just very quickly show you what that would look like for a codex data set. So you see cell segmentation here in this main spatial view. We're going to select one of the tiles of the region that was analyzed in this case. So we're looking at, in this case, thymus data. So we want to select one of the tiles that are sort of more in the center of the region that was analyzed. And you can see the clustering pattern here. So there's a k-means clustering that is selected with a couple of different clusters. This is not a very large number here. You can tell by the number of colors. I think there are about 4 or 5 clusters. And let's take a closer look. So the first thing we can do is, because it's imaging data, it's kind of nicer on the eye to just change the color scheme here. Let's make the whole thing bigger. It's all managed through the UI. And then we can take a look at these results here. So I open up the clustering. There are actually 6 different clusters. Selected cluster number 4, which is this green one here, fairly localized in one corner of the image region. And so we can also, for example, map gene expression levels onto these cells for specific, sorry, these are not gene expression levels. This is something that still needs to be fixed. These are not genes, these are antigens. So we want to just I can show you the abundance of different things in cells. So in this case, it's CD20. And you see this is somewhat localized and kind of correlated with this cluster number 4 that I selected previously. we can also take a closer look at the image data that this information was derived from. So this is an image viewer that can handle any multi-channel data. It doesn't really matter how big these images are. We can create image pyramids and handle really, really large images with many, many channels, as we'll see in a moment. So here, what I did, I basically just modified some of the channel settings. This purple color here corresponds to CD20, and you can see the localization of that on that sort of right-hand side of the image. There are a couple of things, DAPI is in blue, and some other markers that I've selected here. So all of that is happening directly in the browser. You know, once the portal is released, all you need to do is navigate to that page and you can start interacting with the data directly in the browser. Here, I've turned on the gene expression levels once again, just to show you for that selected cluster what that pattern looks like for CD20. Sorry, again, these are not gene expression levels. We are in the process of changing the label to avoid further confusion here. And again, map to the colors of that cluster. So, and here we can make it smaller. In this case, I just selected CD20 and DAPI, so you can see again how that is distributed in the image. Now, let's look a little bit at the provenance, because we want to know where that data came from. As I mentioned, this is a process data set, so there must be a parent data set. And in this case, when we look at this provenance table, again, you can see there's a codex data set, thymus, that this data set was derived from. And what does this look like when we go into a provenance graph view, which we also have integrated here, where you can see, again, the trace basically from the donor all the way to whatever thing you're looking at. And in this case, here I've selected the donor. I can also select the highest level sample that was taken from the donor. In this case, it's the organ, it's the thymus that was taken here. And then I can navigate to that sample. And so now I'm looking at the thymus. And then I can ask, well, what data sets were actually generated from this thymus in addition to that codex data set? And so I can click on this view derived data sets button over here, and I get a table that is filtered. These are data sets filtered by that organ. So here are 4 data sets that were generated from that thymus or based on that thymus. And you see there's a codex data set and there's a single cell RNA-seq data set that was generated as well. And of course, in this case, I navigated to the parent data set for the single cell RNA-seq data. So this is FASTQ data essentially. And what we can do here is we can also look at the assay metadata, for example. We can also download it. So here's just a part of the metadata table. There are probably dozens of different attributes that we capture here. You see the download link at the top right corner. We always have a description for all of the fields in our metadata, so you know exactly, we have exactly the same information in front of you that the submitter of the data had as well. So in this case, you know, see how were the cells enriched or isolated. You can see there's a link to a protocol. Someone mentioned this earlier, we're using protocols.io. Here's the link to the protocol that was used for dissociation of the cells. And we have those links directly from the data portal as well. Okay, so with the remaining time, which I actually don't know how much I have, I want to briefly give you an overview of some of the showcases with additional visualization functionality, data analysis features that we will be incorporating into the data portal in the near future. So this is the first showcase. So here are spleen single cell RNA-seq data sets that were processed using the Salmon pipeline. And then Rahul Satia from the Neurogenome Center ran some algorithms on it to essentially assign cell types to the clusters in the data. So this is functionality that will be in the portal eventually. Right now, this is done externally, and then we just load the results into the portal to visualize them and make them accessible. So here we can scroll down a little bit. And again, here's the visualization, which at this point I should maybe mention, all of the visualizations in the Hubmap data portal right now are powered by a visualization tool called Vitesse. Vitesse is a visual exploration tool for spatial single cell data. It's an open source tool that my group developed specifically for the Hubmap data portal, but that can also be used for anything else. If you go to vitesse.io, there's a whole slew of examples as well as links to GitHub, etc. But coming back here, the visualization, if you see here, again, pretty standard. This is all interactive. I didn't include a video of this one, but I'll show you a video shortly. Shows us, again, a clustering. This is a latent clustering that was done by the Salmon pipeline. In addition, you see an additional set of cell set annotations here, or cell type annotations, that were generated externally and then added to this data set. So let's zoom in here a little bit, enlarge the viewer, and explore this. So when I select these cell type annotations that Rahul Satya's group generated, you see here at the top level, and this important here, this is a hierarchical assignment of cell types. So you see there are hematopoietic cells, there are epithelial cells, and some connective tissue cells. There are hardly any connective tissue cells, a very small cluster. And then we can sort of drill down into that hierarchy to see these assignments with increasing granularity. And so here I zoomed into like a level further down, and you see there are now, the cell types are much more detailed, and you see more colors because there are more clusters. You can see this both in this cell set size bar chart, but you can also see it in the visualization, etc. So we have this information available. You can also select, for example, a couple of cells in here, create your own set of cells, export these cells with their IDs, and use them externally if you would like to do so. Now, this is single cell data. Here is another example of a visualization, in this case, an imaging or multiple imaging assays that we can integrate. Both of these are examples of data that are generated by Hubmap. So these are, this is a multiplex immunofluorescence image of kidney, as well as imaging mass spectrometry data of the same kidney. And these are different slices. This is data generated by Jeff Sprague's group at Vanderbilt. And so let's see, if I play this video, you'll see what we can do here. So let's make it dark, let's make it bigger. And then you can, of course, zoom in. This is what happens when you have interactive visualization tools. You see we have two different image sets here. This is the imaging mass spec data. Here, the smaller grid that's overlaid, lower resolution. And then behind it is the microscopy image. Let's turn off the imaging mass spec data. And we can, with this image viewer in Vitesse, it's very, very flexible. We can do all the color mapping directly in the browser. So this is a WebGL accelerated visualization. We load the raw data directly from TIFF images on the server. We can do color mapping in different ways here. So let's turn to gray. So these are 4 channels. Now the overlaid the imaging mass spec data that we're now bringing back has essentially a whole spectrum of layers in here. So there could be hundreds of layers, but in this case, what we have a couple of mass charge ratios that were picked and that we can now modify. So here I'm just, again, changing the channel, the color mapping for these channels. These channels here, again, mass charge ratios that correspond to different metabolites that were analyzed here in this sample. And you can see this is very flexible, and Vitesse, you can do all of this directly here. And that could be literally dozens or hundreds of different channels if you want that. And so, at the moment, this is mostly to provide quick access to our imaging data, but you can do all sorts of things with this. As you can see, we can really change pretty much anything about how this information is displayed. And in the future, we will, of course, integrate these kind of image data sets with, for example, cell segmentations and other quantitative data that's extracted from the imaging data. Ziv alluded to that earlier, and what can be done if once we set up the analysis pipelines for that. And so with that, I want to thank you for your attention. My group is hiring postdocs, so as well as a senior data curator. So just hit me up on Twitter if you have any interest, questions about our work. If you're interested in data visualization, data curation, or building cool interactive tools, let me know. Thank you.
Thank you, Nils. So I think you probably blew away people, and there's one common great talk and tool. Thanks. There are no questions. So let me ask you a question. So how do you plan to bring forward some of these tools that are still under development? So it's one portal. There are so many tools. It could lead to all sorts of confusion.
Right. So we are planning to, there are different ways in which visualization and visual exploration tools are or will be integrated into the portal. They're sort of, a per data set view. Those are things that I've demonstrated today. In addition to that, what we're planning to do is over the next couple of months or during the remainder of the project period, we're going to build data analysis workspaces that would allow people to combine different data sets and then essentially create a data set of interest and then launch various visualization and visual exploration tools on them. So one of them certainly will be the test. There will also be others that are popular in the community to support various analysis tasks that people might have. I think the need here is for us to build these user interfaces and create a user experience that makes it clear what these different things do, because we're building a lot of tools for different purposes, as you will see in the next talk by Kati Werner. Her group is developing visualization tools and visual exploration tools that do something slightly differently from what we're doing at the moment. But of course, these things are also interacting with each other. You've seen that Ziv is working on something. So we will just make clear what is what in the context of the different analysis tasks and exploration tasks that people are interested in, and organize information accordingly on the portal. And the other question that just came up, I can answer very easily. So the test is built on React. It's very flexible. We've also managed, or others have managed to integrate parts of the tests in Angular and other environments.